If you're enjoying these videos, subscribe. Let YouTube know that I'm doing a good job by subscribing to the channel, plus you'll get notified when I have new videos coming out. And finally, if you're enjoying what I'm doing, give it a like, just click on that thumbs up, and that way YouTube knows that you like it, and they'll pass it along to other people that might enjoy my content as well. I really appreciate it. Thank you again. Let's get started.
Maraming salamat po sir sa inyong mensahe at sa pagbati sa ating mga manonood. Sa ngayon po ay meron po tayong 334 viewers and we are encouraging you to invite your friends at sabay-sabay tayong matuto sa ating programa. At huwag po kalimutan i-like ang aming page at i-share ang ating FB Live. Ngayon naman po ay akin munang ibabahagi ang ating house rules. Una, kung meron po kayong katanungan sa ating resource persons, pakilagay lamang po sa comment box. And please indicate kung para kaninong speaker ang iyong katanungan. At don't forget to mention your name and affiliation. We will be screening your questions and we will raise it later po during the Q&A. Pangalawa, ito po ay webinar on giant clam. Mangyaring ang katanungan po natin ay naayon sa ating paksa. Pangatlo, tayo po ay required mag-fill up ng customer satisfaction feedback form after the end of the webinar para makakuha po tayo ng certificate of participation. We will be posting the links for CSF at the comment box before we end this webinar. Paalala lang po, we will be closing down the link for CSF at 4 p.m. So we hope that you could fill up the form right after this program. So moving on, ang una po nating resource person ay si Dr. Patrick C. Cabaitan. He is an associate professor at the University of the Philippines Diliman Marine Science Institute. He finished his PhD in marine science at the University of Ryukyus, Japan, and was a postdoctoral fellow at the National University of Singapore. His research interests are on community ecology, restoration ecology, giant clam conservation. He is a UP Scientist 1. Sabay-sabay po natin panoorin ang video presentation ni Dr. Patrick, A Home for the Giant Clams, Well-Protected Reefs and Prospects for Socio-Economic Benefits.
Animals that are huggable are utilized to encourage people to support conservation efforts. For instance, this smiling panda. Whale shark is an example of a huggable animal in the marine environment. Another example of an iconic huggable animal is a giant clam. Aside from being huggable, giant clams serve as food and shelter for other marine organisms. Giant clams also provide economic benefits to humans. For instance, giant clams are served as sashimi in restaurants in Palawan, and the smaller and more colorful species of giant clams are utilized for aquariums in many countries. However, because of these economic benefits, many of the giant clam populations are overexploited and got locally extinct. For instance, in the Philippines. According to the late Dr. Edgardo Gomez, giant clams used to be everywhere in the Philippines. For instance, the Scarborough Shoal, or what we call Bajo de Masinloc, used to have fields of giant clams like the ones we have currently in the Silaki Giant Clam Ocean Nursery. In this webinar, I will share information on the giant clam conservation efforts in the Philippines, particularly how we manage to protect and bring back giant clams, and how these efforts provided economic benefits to many coastal communities. I am Patrick, a coral reef ecologist from the Marine Science Institute, University of the Philippines. These conservation efforts were made possible by a collaboration of many people and institutions. Here is the outline of my talk. Why are giant clams important? Why have giant clams declined? What has been done to bring back the giant clam populations? the current status of giant clam conservation, and future efforts. Why are giant clams important? Aside from providing food and habitat to many marine organisms, giant clams filter ocean waters and are a good source of zooxanthellae. Zooxanthellae are dinoflagellates that live in symbiosis with diverse marine invertebrates, including giant clams and corals. The species of symbionts or zooxanthellae may provide information on the adaptive capacity of the host animals towards thermal stress events. Why have giant clams declined? One of the challenges in giant clam conservation is poaching. Giant clams are being poached for food. However, the main cause of the decline in giant clam populations is overharvesting. Recently, even the shells of dead giant clams are illegally collected for use in shell carvings. The bad thing about collecting shells is that people would dig under reef substrates just to be able to get hold of giant clam shells, damaging reef habitats in the process. Another cause of decline and the challenge to giant clam conservation is the recurring thermal stress events or the increasing ocean temperature. For instance, almost every year we observe giant clam bleaching in the giant clam ocean nursery in northwestern Philippines, which causes mortality to giant clams. Although we also observe that some of the bleached clams can recover. What has been done to bring back the giant clam populations, especially the largest giant clam, Tridacna gigas? 8 of the 11 species of giant clams can be found in the Philippines. The bigger species, such as Tridacna gigas and Tridacna derasa, are considered vulnerable. In fact, Tridacna gigas got locally extinct in our country in the 1980s. To bring back the Tridacna gigas populations in the Philippines, the late Dr. Edgardo Gomez started the giant clam conservation programs in the Philippines. 200 kilometers north of Manila, he established the Bolinao Marine Laboratory and the Giant Clam Ocean Nursery, which continue to produce giant clams for conservation and research. Giant clam conservation in the Philippines started in the 1980s with a collaborative research program on giant clam mariculture. With the support of various funding agencies, production of giant clams started. In the 1990s, restocking of giant clams started in many parts of the country. Here is a picture of Dr. Angel Alcala, Dr. Edgardo Gomez, and former President Fidel Ramos restocking giant clams in the Hundred Islands National Park. With the continued support of the Philippine government on research, with colleagues and brilliant students, we continue to advance the giant clam conservation and research, which I will be describing in the succeeding slides. In bringing back the population of giant clams, mariculture and restocking have been done. Reproductively mature giant clams were induced to spawn, and giant clam sperm and eggs were fertilized to produce larvae. Giant clam larvae were reared in tanks for 1 week. Giant clam larvae were then allowed to settle in cement tanks and reared for up to 6 months. Visible juvenile clams were then transferred onto cement substrate so it is easier to transport to the ocean nursery. In the ocean nursery, juvenile clams were placed in cages to protect them from predators. Giant clams that reached a shell length of greater than 20 centimeters are then placed on the substrate to allow them to grow. Depending on the location of the restocking sites, different sizes of giant clams were transported and restocked. Tens of thousands of giant clams, mainly Tridacna gigas, have been restocked in more than 50 reef sites in our country. The different sizes of the blue circles in the map correspond to the numbers of clams restocked in an area. These efforts were successful because of the partnership with many stakeholders, including research institutions, beach resort owners, local government units, and other private institutions. Decades after the initial giant clam restocking programs, we asked what has happened to the restocked giant clams. An ongoing giant clam program funded by the USDP card aims to mainly answer this question. This program is composed of 4 projects implemented by the Marine Science Institute, University of the Philippines, Western Philippines University, and Davao del Norte State College. Findings of these projects will be presented in this webinar. For Project 1, which is headed by myself, we surveyed existing restocking sites and other reef areas in Luzon and Visayas to assess the status of restocked clams, which are mainly Tridacna gigas, and other species of giant clams that are naturally occurring in the wild. Before we did the surveys, we conducted information dissemination activities to enhance the conservation awareness of our partner stakeholders and conducted training workshops to teach LGU staff and fishers how to survey giant clam populations. For the surveys, we mainly recorded the abundance of different species of giant clams in a reef area and measure the sizes of clams, which can tell us whether natural recruitment is taking place in certain reef. Many of the restocked Tridacna gigas clams are already reproductively mature, meaning they are already capable of spawning or releasing eggs and sperm. For instance, a restocked Tridacna gigas clam in Danhugan was observed spawning. Eggs released in the water column get fertilized, then develop into larvae. Larvae get carried away by current and settle and recruit into suitable reef substrate. There are reports that in some of the restocking sites, baby clams or juvenile clams have been recorded. For instance, in Anda, northwestern Philippines, a giant clam recruit was observed near a restocking site. Aside from Anda, we were able to record recruitment of Tridacna gigas in other sites marked by yellow triangles in the map. This indicates that 7 of the 12 project sites have giant clam broodstock that are capable of naturally spawning, and these sites have reef areas that are suitable for giant clam recruitment. Aside from Tridacna gigas, we were able to record different species of giant clams in different abundance in various reef sites. Different species of clams were represented by different colors in the map. For instance, Tridacna gigas is represented by a yellow color, Tridacna derasa orange, and so on. The size of the circles indicates different densities per 10,000 square meters. We noted that well-protected reef areas supported higher diversity and abundance of giant clams. For instance, Apo Reef in Sablayan, Occidental Mindoro. Tridacna crocea, Tridacna maxima, and Tridacna squamosa are common across sites. Rare species such as Tridacna derasa, Tridacna noe, Hippopus, Hippopus porcellanus are still present at certain sites, for instance in Apo Reef and in Calaguas. In this giant clam research program, we were also able to assess the types of symbionts present in giant clams in different sites. Preliminary analysis indicates that Tridacna gigas and Tridacna crocea giant clams in each of the sites had distinct symbiont community structure as represented by different color combinations in different study sites. Tridacna crocea had higher diversity of symbionts than Tridacna gigas. Combining all the data of symbiont community structure in different giant clam species found in different sites, here is the pattern of relative abundance of the three most common genera of giant clam symbionts. This would provide insights on how giant clams in different sites may respond to thermal stress, as some symbiont species, for instance Durosdinium, represented by a red color, are more resilient to thermal stress. In the succeeding slides, I will show present and future efforts that may provide socio-economic benefits to coastal communities. In some of the restocking sites, for instance in Samal and Camiguin, giant clam conservation has provided more socio-economic benefits to the stewards of giant clams. Giant clam restocking sites in these areas have become a tourist attraction, which provided added income to fishers and other stakeholders. The success of this effort is mainly because of the collaboration among multiple stakeholders such as people's organization, local government unit, barangay, the academic institution that helped initiate the establishment of the restocking site, specifically the Davao del Norte State College. By tour guiding, the association was able to acquire a revenue of around 2 million pesos for only 3 years from the start of the operation. Updates of this effort will be presented by Dr. Girley Gumanao in this webinar. Aside from Camiguin and Samal, here are the other giant clam restocking sites that become a tourist attraction. Bolinao, Hundred Islands Natural Park, Batangas, Palawan, and Sagay in Negros. However, these sites are not as successful yet as the restocking sites in Camiguin and Samal in terms of obtaining revenues from ecotourism efforts using giant clams as attractions. Future efforts should include establishing guidelines for operating ecotourism activities with giant clams as the main attraction. Another benefit in restocking giant clams is that it would help enhance reef fish communities in degraded patch reefs. An increase in fish abundance and species richness were observed relative to control and to shell treatment 3 months after restocking of giant clams. After knowing that restocked clams can enhance fish communities in degraded reefs, we asked, has giant clam restocking had a positive effect on the fish catch of fishers from nearby coastal communities? To answer this question, we conducted a fishing experiment using fish traps to see whether more fish would be caught in a giant clam restocking site relative to the other sites with no giant clams, such as seagrass, lagoon, and a reef area. Here's a picture of the clam site with a high abundance of fish. In this study, we also conducted fish visual census to quantify fish communities in different sites. Results of experimental fishing showed that more fish traps at the giant clam site contained fish, although fish counts per trap were lower in the clam site. More fish individuals were caught per trap at the reef area. However, there were higher species richness in fish traps at the clam site compared to the other sites. Based on interviews, fishers reported that they were able to catch more fish in clam site, and they perceived that the increase in fish catch was because of the presence of giant clams. One of the main reasons for the success of giant clam conservation is the cooperation of fishers and local organizations. In this site, giant clam recruits, represented by black circles, have been observed near the restocking site, which is inside Cabaitan Marine Protected Area, up to as far as 2 kilometers from the restocking site. Fishers have been reporting and tagging locations of giant clam recruits, which provides information on the potential dispersal of giant clam larvae from the broodstock populations inside Cantaan MPA. This data supports the idea and purpose of establishing marine protected areas wherein animals such as fish or giant clams, for this matter, inside a marine protected area are not affected by fishing while they grow, reproduce, and provide recruits into the outside of a marine protected area for harvesting. Although harvesting giant clams currently is solely for conservation and research purposes only. Future giant clam conservation efforts should continue to monitor giant clam recruitment inside and outside marine protected areas, so we'll have a full picture of how much recruits can a giant clam broodstock population provide outside marine protected areas in other sites. We have observed that the giant clam broodstock population inside the Cantaan Marine Protected Area was only able to provide at least 5 giant clam recruits outside the protected area over a period of at least 3 years. This information suggests that if we need more giant clams to expand restocking efforts, there is still a need to culture and rear giant clams. One way to do this is by establishing demonstration sites for community-based farming in suitable reef areas and in areas where support and cooperation from fishers and local government units are present. This will minimize the cost and effort in transporting giant clams from the main source in Bolinao, Pangasinan to different restocking sites. With the ecological functions and socio-economic benefits provided by the giant clams, we hope to continue our efforts in giant clam conservation and research by involving and enhancing the capacity of other local and regional institutions in giant clam culture and restocking. There are now 8 functioning hatcheries in the Philippines that can help in continuing the efforts in giant clam culture and restocking. And we hope that the success in conserving giant clams through collaboration among multiple stakeholders can be replicated in a wider regional scale by having a multinational collaboration in the conservation of giant clams and reef habitats. To end this talk, I would like to highlight the following. We should continue monitoring reef sites and giant clam populations, especially that we do not know yet how much recruits the other broodstock populations are providing in other reef sites. We should provide better protection in reef areas, as we have seen that giant clam broodstock populations are able to provide recruits to adjacent reef areas. We develop hatcheries and capacity for cultural local wild stocks in other strategic locations. Restock giant clams at well-selected sites. Cooperation between local government agencies and coastal communities is important for success in giant clam conservation efforts. Lastly, I would like to thank our partners, the fishers, local and provincial government units, academe, and other stakeholders for making the giant clam conservation efforts successful. We look forward to working with you again when we expand our giant clam conservation efforts in the near future. Thank you.
Thank you, Dr. Patrick, for that very informative presentation. Nakita po natin sa presentation ni Dr. Patrick na napakadami po nating taklobo. But here comes the poaching, over-harvesting na naging dahilan ng unti-unting pag-uubos nito. Nagpapasalamat po tayo sa mga nagmamalasakit na mapangalagaan ang ating karagatan. Thank you, Sir Patrick, for sharing the initiatives of the research team and the efforts in the management and conservation of the giant clam resources in the country. So let's learn more about it po to our next 3 resource person pa po. So kung meron po kayong katanungan kay Dr. Cabaitan, maaari nyo pong ilagay sa comment box. So before we proceed to our next presentation po, atin po muna batiin ang iilan sa ating participants. So magandang umaga po sa ating manonood from MSU Sulu, Sir Frads Manyalas. Meron po tayong viewers from MSU IIT, Ma'am Tampus. From Cavite State University, Ma'am Herrera. Magandang umaga po sa inyo. Meron po tayong viewer from College of Fisheries Laguna, State Polytechnic University. Magandang umaga po. Magandang umaga din po from LGU Oroquita, City of Enro, Oroquita City. Kay Ma'am Emperio. Meron din po tayong manonood na OFW from Aklan. Magandang umaga po, Sir Inamak. So patuloy nyo lang pong i-share yung ating FB Live. At salamat po sa inyong panonood. Our next speaker po is Dr. Roger G. Dolorosa. He is a professor at the Western Philippines University. He finished his PhD at the School of Environmental Sciences, University of East Anglia, United Kingdom. His research interests are on marine biology, ecology, and conservation. He is the current editor-in-chief of the Palawan Scientist, the official publication of the Western Philippines University. Let's watch the video presentation of our second speaker, Dr. Dolorosa, on evaluating the status of giant clam in Palawan.
Good morning, everyone. On behalf of my co-authors, I would like to present our findings for this research project evaluating the status of giant clams in Palawan. Giant clams are the largest living bivalves. The largest species, Tridacna gigas, can reach up to 1.3 meters, while the smallest, Tridacna crocea, could be about 15 centimeters in shell length. There are 8 species of giant clams in the Philippines, and all of these also occurred within the waters of Palawan. Giant clams provide a wide array of ecological and economic services. Here we can see that a giant clam serves as important substrate to a wide array of marine organisms. They also serve as food to numerous organisms, and they also help increase the complexity of the reef. As filter feeder, giant clams can help reduce the sediment in the water column, thereby promoting a healthy coral reef environment. In spite of its ecological and economic uses, giant clams are unsustainably harvested, and for that reason, all giant clam species are listed in the Appendix 2 of CITES, which means that they are not threatened with extinction, but they may become if trade is uncontrolled. In the Philippines, all giant clams are considered threatened, which means that harvesting and trade is a crime and is punishable by law. It's been 20 years since some of these laws were enacted, and many conservation actions were done to protect the giant clams. However, there is no updated information about the status of these species in Palawan, and because of that, we conducted the study with the following objectives. We looked into the recruitment and abundance of giant clams in 4 study sites such as Taytay, Port Barton, Binduyan, and Rasa Island. However, with the support of other organizations, we were able to visit other areas as indicated by the green circles. With the use of social media and help of many individuals, we were able to cover a much wider area when it comes to information about the presence of the largest giant clam species, Tridacna gigas. In addition, we also conducted several information and education campaigns in various areas of Palawan. To collect the data, we used the phototransect method to determine the percent hard coral cover. We took photos of the substrate along the transect and determined the percent coral cover using CPCE. And then, giant clams found 2.5 meters on both sides of the transect were photographed with a size reference beside it. Here we have a video of a researcher while doing a phototransect and while taking a picture of giant clam with a measuring rod as size reference. These are the transects and areas covered at each study site. We were able to survey 29 transects in Port Barton and 3 transects in Rojas. In total, we surveyed 57 transects covering 12,375 square meters. As for our results, in terms of the coral cover, Port Barton had the highest at 31.5%. This was followed by Rasa Island and the lowest was in Rojas. These low coral covers could be attributed to the outbreak of crown of thorns, starfish, and the frequent bleaching event. These values fall within category D or poor coral cover except for Port Barton, which fall within C or fair coral cover. In terms of species richness along the transects, Rojas had the highest number of giant clam species, which was 5, and the low number of species at each study site is an indication that some species of giant clams are difficult to find, possibly due to overharvesting. In terms of density of giant clams, Taytay had the highest at almost 3 individuals per 100 square meters. Rojas comes next at 2 individuals per 100 square meters, and other sites had less than 1 giant clam per 100 square meter. These densities are not related to the health of the reef, considering that Port Barton had the highest coral cover and Rojas had the lowest. These densities are very low compared to densities being reported in well-managed reserves. The lesser number of adult clams is an indication of overharvesting. However, the presence of juvenile clams at each site is a good indication that settlement is going on. When effectively managed, the populations of giant clams in these sites are expected to recover in the next few years. As for Tridacna gigas, we recorded a total of 19 individuals. 7 and 6 individuals were documented in Tubataha Reefs Natural Park and Rita Island, respectively. The clams in Dos Palmas and Rita Island were induced to spawn with the help of UPMSI and funding support from the Malampaya Foundation. The juveniles we produced were restocked in priority areas of the Malampaya Foundation Incorporated. In terms of information and education campaign, we presented the results of the study to the LGU of Taytay, to the LGU of Port Barton, the LGU of Binduyan, and the Rasa Island Wildlife Sanctuary Protected Area Management Board. We also conducted IEC outside our study sites. This picture was taken during an IEC in Brooks Point National High School. This was in Puerto Princesa City, also in Puerto Princesa, but our audience were faculty members from a state college in Zamboanga. This was during the Catala Foundation held in Puerto Princesa City, and we also did an IEC among students and fishermen in Port Barton. So, moving forward, in order to hasten the recovery of the lost populations, there is a need to sustain the ongoing giant clam restocking project. So far, a few hundreds Tridacna squamosa and Tridacna gigas were restocked in 6 localities in Palawan with funding support from the Malampaya Foundation Incorporated. We observed that some conservation initiative involving the aggregation of giant clams, and to ensure the full benefits derived from this project and avoid its use as front for any illegal activities, the LGU, in collaboration with academe and stakeholders, should create a local policy on this kind of initiative. In this video, we could see that those clams were placed on top of a living coral. And lastly, we need to become more vigilant because recently there were confiscations of large volumes of giant clam shells in Palawan. Giant clams are used as alternative for elephant ivory, and in April 16 alone, giant clam shells worth more than 1.2 billion pesos were confiscated in the municipality of Rojas. With that, I would like to thank the DOST PICAARD, the UPMSI, Atelier Aquatic, WWF Philippines, the municipalities of Taytay and Nara, Barangays Binduyan and Port Barton, as well as the Malampaya Foundation for all the support. Thank you very much.
Maraming salamat po sa pagbabahagi ng inyong pananaliksik, Dr. Dolorosa, regarding the status of giant clam in Palawan. So in this conduct of this study, ang dami pong tumulong sa research team. Kung makikita natin, andyan po yung private organization, LGUs, and researchers to cover a wider area and to get more data. So it showed that they are united in the conservation of our marine resources. Sana po ay marami pang tumulong para mapangalagaan at mapanatili na mayaman ang ating karagatan. So maaari nyo na pong ilagay ang ating questions para kay Dr. Dolorosa sa ating comment box. So moving forward, our third resource speaker is Dr. Girley S. Gumanao. She is a professor at the Davao del Norte State College and the BP for Academics and Research. She finished her PhD biology from Davao del Norte State College. Her research interests include coral reef ecology and conservation. Dr. Gumanao leads the giant clam conservation in Samal Island, southern Philippines, known as Taclobo Tours, that drives environmental advocacy campaign, provides livelihood opportunities to local communities, and sustain giant clam conservation efforts in partnership with academe, LGU, and people's organization. For our third presentation, here is Dr. Gumanao on her research, Giant Clam Conservation Brightspot in Mindanao, a hope to return the few giants.
A pleasant day to everyone. This is Girley S. Gumanao of the Davao del Norte State College, Panabo City, Philippines, to present the topic Giant Clam Conservation Bright Spots in Mindanao, Hope for the Giants. This presentation highlights the outputs of Project 3, which is under the DOST PICAARD funded program Assessing the Status of Giant Clams and Advancing Culture Techniques, led by UP Marine Science Institute. Giant clams are the largest living bivalves that live in close association with the reefs throughout the Indo-Pacific. They are ecologically important as biomass and predators and scavengers and as substrates of epibionts. They improve reefs' structural complexity and provides habitats to fish and marine organisms, thus enhances biodiversity in the area. Unfortunately, harvesting for local consumption, export, and shellcraft have led to population declines worldwide, including the country Philippines. A nationwide giant clam restocking program led by Dr. Edgardo D. Gomez was implemented in over 40 sites in the Philippines, including the sites in Mindanao. After 2 decades, this project intends to find out if the restocking efforts were effective in the conservation of giants. Specifically, the project intends to survey giant clam recruitment, assess giant clam biodiversity and abundance, and conduct information dissemination and skills transference. The study sites are located in Mindanao. It is the second largest island in the Philippines in the southern part of the archipelago. Sites for biodiversity assessments were in Tawi-Tawi and Island Garden City of Samal. Tawi-Tawi is the southernmost province of the country with over 300 islands. 4 sites were covered in the area. Igacos is located almost in the middle of Davao Gulf. It has 2 islands, Samal and Talikod Islands. 10 sites were surveyed in the area, mostly marine protected areas. Restocking sites in northern Mindanao included only 1 study site each. Prior to the conduct of the study, we secured permits from the LGUs and concerned national agencies. We also partnered with the local organizations, including the HEIs in the locality. Sampling was done using belt transect at 1.4 to 16.4 meters depth. Giant clams along the transects were identified, measured, photographed, and geotagged. To triangulate the results, we also conducted focus group discussions, and we surveyed the presence of giant clam shells along the shores. For Samal restocking site, all giant clam species were identified, measured, photographed, and tagged with permanent markers to establish monitoring sites. Due to the limitations of the pandemic, the species in the restocking sites in northern Mindanao were studied with the use of data from snorkeling and photo documents that were gathered during the preliminary survey. For this presentation, the recruitment data will be presented using the recent secondary data. For information dissemination and skills transference, we utilize the outputs of the project. A total of 8 species were found in Mindanao. This constitutes 100% of the species found in the Philippines. These species include the restock Tridacna gigas and Tridacna dirasa, the common species Tridacna squamosa and Tridacna maxima, other species such as Tridacna crocea, Hippopus hippopus, and Hippopus porcellanus. The newly resurrected species Tridacna noe is also found in Mindanao. Tridacna gigas was found in most of the sites except for Tawi-Tawi. However, it is suspected that it is present in the area due to the sightings reported by local communities. There were 3 species common in all sites. Hippopus species were absent on the island. The density of giant clams in Igacos was 338 individuals per hectare and 587 individuals per hectare in Tawi-Tawi. In Tawi-Tawi, Tridacna crocea and
Susulyut-sulyut and Tridacna maxima manangkaykima were among the abundant species in the area. Mean size shows that there were lots of recruits or small-sized individuals of this species in Tawi-Tawi, even in fishing areas. These substrates were suitable to the existence of these species. In the island garden city of Samal, Tridacna maxima and Tridacna squamosa, known as Taclubo, were among the abundant species in the site. Based on the maximum size, there were individuals that exceeded the maximum size reported for this species. It should be noted that they are protected by MPA in Samal Island. These were the substrates in the site. Tridacna crocea, the boring clam, was perceived to be widely distributed in the past, but recently it was perceived to be locally extinct. This coincides with the observations in the field sampling. The occurrence of giant clamshells along the shores was studied to account for the species that may have existed in the past. 5 species were found, and surprisingly, Hippopus hippopus, the kukong kabayo, had the most abundant shells. It is sad to note that Hippopus hippopus were not found in natural habitats. This indicates that the species existed in the past, and its absence in natural habitat suggests local extinction. This observation is also true to Tridacna crocea. However, few individuals were still observed as turnovers by local fishers. The use of giant clamshells were also observed and reported. The observation suggests the strong dependence of humans to the giant clam species. Such practices and beliefs creates challenges and threats to the giant clam species. The restocking site in Samal Island is a marine protected area managed by the Davao del Norte State College in collaboration with the community and LGU. It is a home of more than 2,000 giant clams. There are 6 species, including the restocked Tridacna gigas and Tridacna dirasa in the site. The last 2 species were recently added as turnovers from concerned coastal communities. Tridacna gigas is the most abundant and the largest species in the area. The largest T. gigas measures 98 cm, which is almost a meter in size. Recruits of T. gigas were found during surveys and through turnovers by concerned fishers. Recently, natural spawning of T. gigas was observed in the restocking site few days before the full moon. It was a mass spawning event, and giant clams have released both eggs and sperms that lasted for days. Additionally, T. dirasa recruits were also found near the restocking site. As presented, 6 species are present in Igacos, where Tridacna squamosa and Tridacna max are the most abundant species. As shown, they are widely distributed around the island, while the other 4 species, including the Tridacna gigas and Tridacna dirasa recruits, were only confined in the western side of the island where the restocking site is located. From the restocking site, recruits of T. gigas and T. dirasa were found at 3.8 kilometers north and 11.4 kilometers south directions. This coincides with the flow of the north and the southbound water currents. T. gigas recruits were also found 6.4 kilometers across the smaller Talikod Island. The mean size of T. gigas recruits is 14.9 cm and 9.4 cm for Tridacna dirasa. The presence of recruits indicates the spillover effect of the MPA and the biological impact of giant clam
restocking in Samal Island.
Giant clam conservation in Igacos is a bright spot. It is the site of Taclobo Tours, a giant clam ecotourism project. It features the huggable gigacos, named after Tridacna gigas and Igacos. While earning income from the tour, local fishers who were formerly giant clam poachers are now strongly advocating for giant clam conservation in MPAs. Indeed, giant clams has truly transformed the lives of the local fishers. Taclobo Tours manifest the social and economic impact of giant clams restocking in southern Mindanao. Likewise, the restocking site in Camiguin managed by the Cantaan Centennial Multipurpose Cooperative have also shown both biological and socio-economic impact. It is also famous for giant clam ecotourism even earlier than the Taclobo Tours. It has successfully protected viable spawning populations that were able to produce recruits outside the restocking sites. This is clearly shown in this published article. The project developed giant clam identification manual with the use of shell morphology and giant clam mantle. It was used in the capability building of its partners, MSU Tawi-Tawi, BFAR 11, and other stakeholders. The status of giant clams in Mindanao calls for a strong advocacy. A logo was created with the slogan, "Ang Taclobo bililhon sa kadagatan, atong bantayan," which means, giant clams are valuable, let us protect them. The logo was etched to all advocacy materials such as store awnings and mugs, and were distributed to communities and fishers with a strong confidence that this will inspire the fishers and stakeholders to protect the prohibited species. With this, we can conclude that Mindanao represents all giant clam species in the Philippines with higher densities in Tawi-Tawi. Restocking sites serve as viable source of spawning populations of restocked species, and suitable substrates are provided by adjacent reefs. Protected areas in Samal maintains broodstocks of local species, while Tawi-Tawi has natural stocks of Hippopus species. There is strong commitment of local organizations, LGUs, in collaboration with fishers to sustain giant clam conservation in Mindanao. Restocking sites could serve as demonstration area for sustainable ecotourism. There are still threats to giant clams in Mindanao due to strong dependence of communities to giant clam species. Hippopus hippopus is threatened by local extinction in Samal Island.
The following are our recommendations. Strengthen marine protected areas as refuge of giant clams. Continuous advocacy due to continued harvesting and collection. Incorporate findings of the study in the advocacy materials of ecotourism sites, such as inclusion of additional species in the tour, closed season for tourism during spawning season. Formulate local ordinance to support national laws on the protection of giant clam species. Establish restocking sites through local giant clam hatcheries and ocean nurseries in areas with functional MPAs and good community support. To end, I would like to thank and acknowledge the following agencies and persons who were with us during the implementation of the project. To all, thank you very much for listening.
Thank you, Dr. Gumana, for that very interesting and informative presentation. Kung pansin po natin, there's a lot of restocking activities na of giant clams conducted in different areas in Luzon, Visayas, and Mindanao. Marami na po tayong nagawang pananaliksik at nakikita na po natin yung progress and positive impact nito sa ating karagatan. So despite many challenges and threats, we believe that we can achieve our goals if we are united in this conservation advocacy. Once again, thank you po, Dr. Gumanao and to your research team. Kung meron po kayong katanungan para kay Dr. Gumanao, maaari na pong ilagay sa ating comment box. So bago po tayo magpatuloy sa next presentation natin, atin po munang batiin ang ilan sa ating participants. Magandang umaga po from Negros. Mayroon po tayong participants from Negros Oriental State University, SSU Cotabato, Pampanga State Agricultural University, Samar State University College of Fisheries, Bicol University, Tabaco Campus from DOSCST Main Campus, and from BFAR 4A, SPAMAS, DLSU, and BFAR and BFFTC. Magandang umaga po sa inyong lahat. So our fourth speaker is Dr. Cecillia Conaco. She is an associate professor at the University of the Philippines Marine Science Institute and head of Marine Molecular Biology Laboratory. She obtained a PhD in molecular and cellular biology from Stony Brook University in New York and was a postdoctoral fellow at the University of California, Santa Barbara. Her research interests include gene expression dynamics, development and evolution, and marine symbiosis. She served as Deputy Director for the Bolinao Marine Laboratory in Pangasinan from 2015 to 2019. She contributed to revitalization of BML Giant Clam Conservation Program through the introduction of the BML Adapt a Clam program, organization of giant clam training workshops, and initiation of the Silaki Giant Clam Marine Park in collaboration with Bolinao Municipal Government. Here is the Dr. Conaco on her presentation, Big Things Come from Small Beginnings, Gene Expression During Early Development of Giant Clams.
Good morning. My name is Cecillia Conaco, and today I will be telling you all about our work on Giant Clam Project 4. Most of the findings I will be presenting here were generated by Kiana Tan, the research associate on this project. Good morning. In the previous talks, my collaborators on this program described the results of their surveys at different reefs throughout the Philippines. Some of those reefs were restocking areas for giant clams in the 1990s. Giant clams for restocking are usually grown to larger size in an ocean nursery. Before this, they were reared for several months at a hatchery where they were produced through induced spawning. Good morning. It takes a lot of effort to grow giant clams. First, we prepare the broodstock. Then, we induce spawning. We collect gametes and mix them together. We fertilize the eggs. We check on larval development. And then we rear the larvae until they can be settled onto substrates. These hatchery culture techniques that we still use today were developed in the 1980s at the start of the Giant Clam Conservation Program in the Philippines. Good morning. The journey from egg to giant clam takes a very long time. Tridacnagigas start off as microscopic larvae. They first become visible on the substrate at around 1 to 3 months of age, and after 1 year, they can grow to about 5 centimeters in size. However, it will take 2 to 3 years before these clams can produce male gametes, and about 8 to 10 years before they produce female gametes. Good morning. All giant clams start as tiny larvae and pass through several key developmental stages. Within the first day after an egg is fertilized, the free-swimming trochophore stage appears. After a day or two, the trochophore transitions into the veliger stage and begins feeding. After about a week, the larvae become pediveligers, which alternate between swimming in the water column and exploring the substrate. After this stage, the larvae begin metamorphosis, where they lose their swimming capacity and juvenile organs begin to develop. At around the same time, dinoflagellates begin to colonize the mantle tissues of the clam, where they will proliferate to become the main nutritional source of the organism, helping it to grow to larger size as they mature into adults. In giant clam aquaculture, high mortality generally follows developmental transitions. As you can see in this sample taken from our culture, there are many empty or dead larvae alongside live larvae. Though a low survival rate is generally expected for broadcast spawners, which release millions of gametes, this can also be exacerbated by stress that is experienced by the larvae while they are in culture, especially if they take too long before they can find a good settlement substrate. We typically observe survival of less than 5% after 6 to 8 days of culture, and studies have shown that survival can be even less than 0.5% at 6 months. Thus, the development of technologies and strategies to better manage culture through the critical early stages are needed. A better understanding of the basic biology of larval development is important so that we can design interventions that may improve survival in culture, which will in turn increase giant clam yields to support local restocking and conservation efforts. To gain information on the development of giant clams, we used the following strategy. First, we conducted spawning and culture of 2 species of giant clams, Tridacna gigas and Hippopus hippopus. Then we collected samples from 6 developmental stages from each species. We then extracted RNA and performed next-generation sequencing. And finally, we performed gene expression analysis to identify the genes that are active during giant clam development. Good morning. The data that we get from next-generation sequencing consists of millions of short fragments of DNA. We assemble these fragments together to identify the transcripts or genes that they belong to. Then we determine at what developmental stage each of these genes is most highly expressed. After this, we assign functions to the genes, and then we assess which functions are enriched at specific stages. From our data for the 2 giant clam species, we identified 3 major gene expression patterns. The egg genes are dominated by functions related to the cell cycle, stem cell maintenance, DNA repair, DNA replication, and others. Larval genes, which are expressed in the trochophore and veliger stages, are mostly involved in tissue morphogenesis, ectoderm formation, endoderm formation, and patterning of the organism. Settlement genes, on the other hand, are expressed most highly in the pediveliger and early juvenile stages, and they are mainly involved in animal organ development, as well as regulation of growth, cell differentiation, cell matrix adhesion, response to hormone and catecholamine metabolism. We were particularly interested in catecholamine metabolism because other studies in bivalves have shown that catecholamines are strong inducers of settlement and metamorphosis. Catecholamines are hormones like dopamine, norepinephrine, and epinephrine. They are produced by various enzymes in the catecholamine biosynthesis pathway. These molecules bind to cell surface receptors to send signals into the cell. In giant clams, we found that the enzymes that produce catecholamines, as well as their specific receptors, are highly expressed in the pediveliger larvae, which are just about ready to settle. We hypothesized that binding of catecholamines to their receptors will signal the larvae to begin the changes that are required for settlement and metamorphosis. To test our hypothesis, we conducted settlement assays. We found that addition of the catecholamine epinephrine was able to significantly enhance settlement rates when compared to the controls. As you can see in this video, the larvae in the beaker with epinephrine begin to settle quickly to the bottom, while those in the beaker containing only seawater remain actively swimming around. Addition of epinephrine also enhanced metamorphosis rate, with 59% of the larvae being at the intermediate and complete metamorphosis stages within 72 hours of exposure. Metamorphosis is indicated by shedding of the velum, as you can see in this video. Metamorphosis was observed in some individuals as early as 3 hours after initiation of the experiment. These findings suggest that epinephrine may indeed be useful in giant clam aquaculture for the purpose of enhancing settlement and metamorphosis. This may eventually translate to better survival and higher yield. There are many other interesting findings to be discovered from our data. For example, we found that genes involved in shell biomineralization are expressed as early as the trochophore stage, where they may be involved in the production of the embryonic or larval shell that we observe in veligers. Stress response genes, such as heat shock proteins, hypoxia-inducible factor, and antioxidant gene families, are most highly expressed in the veliger and pediveliger stages. These genes may help the larvae maintain homeostasis as they undergo the process of metamorphosis. Symbiont genes involved in photosynthesis, carbon dioxide transport and metabolism, and nitrate assimilation increased in expression towards the later developmental stages, corresponding to establishment of symbiosis. In summary, we have learned through this project that gene expression during the early life of giant clams is highly dynamic. We have also learned that catecholamine signaling, particularly through epinephrine, may be a way to enhance giant clam settlement and metamorphosis. And finally, we have shown the utility of transcriptome sequencing as a way for us to identify giant clam genes and to determine when these genes are expressed during development. Good morning. Through this project, we developed extensive sequence resources for Tridacna gigas and Hippopus hippopus. We have also generated sequence data for 5 other species of giant clams. But having these resources on hand is really just the beginning. Further studies will be needed to look at all this information and to translate it into better understanding of our giant clams. Future applications for these sequence resources include further investigations on symbiosis, calcification, and the stress response of giant clams. These genetic information can also be used to assess resilience and other desirable characteristics, as well as to develop markers for monitoring giant clam health or to trace giant clam populations. Findings from such studies will contribute to the improvement of culture technology, support giant clam conservation efforts, and provide critical information for management. Coupled with continued site monitoring data, these studies will bring us closer to our goal of making sustainable use of giant clam resources for activities such as ecotourism or even community-based farming, which will benefit coastal communities. To end this talk, I would like to acknowledge the following people who assisted us in this project: the Bolinao Marine Laboratory, where we conducted our experiments, and funding support from DOST Picard.
Thank you, Dr. Conaco, for sharing the results of your study. If isipin natin yung journey ng giant clam from egg to adult, every life stage is very beautiful, no? And that is something that we should protect given its functions and benefits to our environment and to our economy. So, if you have comments po for Dr. Conaco's presentation, maaari pong ilagay sa comment box. So, thank you po sa apat nating resource person for sharing your researches, initiatives towards conservation of the giant clam in the country. So this time we'll be picking up and try to provide answers to your questions from the comment box. So ito po ang ating first question. Ang atin pong first question ay para kay Dr. Patrick. So why is it important to survey and monitor giant clam recruits? Okay, Sir Patrick.
Hello. Okay, to answer that question, now we've been monitoring abundance of giant clams, but we do not know what happens to them after a few weeks, few months, or a few years. No, we have to know their recruitment, and aside from recruitment, we need to know whether they are growing and then reproducing. So if we know these information, we can tell that certain population can uh, sustainably reproduce and increase by itself in an area. So yun po ang importance ng pag-identify or pag-measure or pag-quantify ng recruitment. So as shown in the earlier slide, for instance in Camiguin, if you have a protected area, you can see that the broodstock that are protected in that area can produce larvae and allow settlement or recruitment of giant clams outside the marine protected area. So if we continue to do this, we may be able to have like a sustainable way of reproducing giant clams. Although with this, we've seen that there were only around 5 recruits in over a 3-year period of monitoring. So that means they still need assistance from us. Like for instance, we still need to culture giant clams. One suggestion was to involve communities like coastal communities to do community-based farming to increase the stocks of giant clams.
Thank you, sir. So ito po yung second question natin. Ang question po ay para kay Dr. Gumanao. Do you know the regional number of T gigas placed by UPMSI in restocking sites in Mindanao? How do you compare it with the current status of giant clams in the restocking sites?
Hello, good morning.
Good morning, ma'am.
So, for the case, I can only tell about the restocking site in southern Mindanao.
Question natin.
Originally in the Samal Island restocking site, there were sub-adults and adults that were placed for Tridac na Gigas and Tridac na Derasa. So for Tridac na Derasa, originally there were 1,092 individuals for large and sub-adults. And there were also small ones, the juveniles, more than 2,000. It is written that they were juveniles and they were placed in southern Mindanao. I guess there are also other areas in southern Mindanao like Tawi-Tawi that I cannot trace if it's just specific for Samal Island. In our case, I can say that the large ones, like 1,092 individuals were placed in our sites. And currently, 77% of this are left in our area. We have more than 800 individuals with almost 1 meter in size. For the Tridac na Giga, at Tridac na Derasa, there were 520 individuals originally placed. And now we have 81% of these individuals left, and they're already spawning or matured populations. There were also like Tridacna squamosa that were spawned by, I think, Dr. Patrick and Dr. Gomez before. Source is from Samal Island, and they flew to MSI for rearing, and then they fly back to Samal Island. And I think there were lots of them, more than 21,000. However, they were part of the restocking efforts conducted by DNSC, and they were distributed throughout the island. That was in collaboration with Florendo Foundation. That's the time of Dr. Estrellada. So I think I can say that this is the status now. We have like 77% left for Tigigas, 81% more for Tridacna dorasa. These are the large individuals. And I can say that they have increased in size. No, just imagine when they were restocked, they're just like 20 cm, and now they're all almost a meter. And I can say that there are already spawning populations, and now they can replenish depleted stocks in the wild, as we have shown in the recruits. We have actually 43 recruits for Tigigas, and we have 15 recruits for Tidirasa. And that's already very good. That's why I can say that the giant clam species that were restocked in Samal Island restocking sites is the hope for our very few giants 20 years ago.
Thank you, Ma'am. Thank you, Ma'am Girley. I guess nasa protected area naman po sila, and naka-establish na po yung team or yung concerned agency po para maprotektahan po yung ating natural resources. So let's proceed po sa third question. So our third question ay para po kay Dr. Dolorosa. Aside from over-harvesting, what do you think are the other major contributors to the disappearance of giant clams?
Good morning, and thank you for that question. Naririnig po ba?
Yes, sir. Yes, po. We can hear you.
Aside from overharvesting, I think the other factors which contributes to the disappearance of giant clams in the wild is habitat destruction. Usually, habitat destruction brought about by destructive fishing methods such as dynamite and cyanide fishing. Most of us are probably aware that giant clams occurred in reef areas, in coral reef areas. And if you destroy the habitat, you are not just destroying the fish, but also the corals and also the giant clams. In the Philippines, only about 5% of our coral reefs are in excellent condition. That is because of habitat degradation. So that would also reflect the population or the condition of giant clams in the wild. Another problem could be the level of management of our marine protected areas. So although we have plenty of MPAs now, but I think only about a small percentage are well managed. Maybe about 25% or even lesser than that. So what we have noticed is that even in some marine protected areas or in sanctuaries, the populations of giant clams are quite low. So if these marine protected areas could be managed appropriately, we could, help restore the lost populations of the giant clams. I'm not sure if I did answer the question. Thank you.
Thank you for that, sir. So let's move on to our fourth question. This question is for Dr. Conaco. How does epinephrine treatment affect the larvae in terms of mortality?
Hi. Good morning. Thank you for that question. So in terms of larval mortality, we found that exposing the larvae to epinephrine is not toxic. So the larvae are okay. They settle very quickly to the substrate. However, we're still under the process now of checking survival after settlement, which is a very important stage, and that takes quite a while. Um, but in initial studies or initial counts that we've had, medyo mataas naman yung survival with epinephrine treated um, larvae. One of the things we're looking forward to is seeing kung, because they settle very quickly and metamorphose rather quickly with epinephrine, we're hoping that we would get more yield later months of culture, just because mas malakas yung larvae siguro when they settle. Hindi sila masyadong matagal na exposed to stressful conditions in culture. But really, all of this is still a work in progress, and we're hoping to have more updates in the very near future. Thank you.
Thank you, ma'am. So hopefully po, you can share with us yung further result po ng study nyo. Ano? Question po? So ito po yung fifth question po natin from Philip Shu. The beginning what happened during the covering period until now. Wandering in Masinloc, the fisherman's wharf has no Taclobo hub, not for tourists except eating the Taclobo from Scarborough Shoal. But this is better to start once again. So I was in detail before that the office, sorry po medyo hindi abot ng screen ko. So I was detailed before at the office of Sec Mitra. What is given to me Zambales, then Nara, Palawan. So we pick up shell clam at Bito Cruz beside the Philippine Navy before Imelda Marcos constructed the cultural center of the Philippines. So ano po pinaka question is from 1980, the beginning what happened during the covering period until now. So yun po. So anyone from the group po, yung may pwede ko yung may i-share po about this. Kasi po sinasabi natin 2 decades na din yung conservation efforts po na ginawa ng Pilipinas para po dito sa pag-restore and pag-manage po ng giant clam. So ano po masasabi po ninyo dito sa ating comments? Sige po.
JP, I can add, I can provide information.
Okay po, sir. Sige po.
Yeah, as mentioned in a lot of presentations, there are now a lot of giant clams restocked in different sites. But since you mentioned about Masinloc and Scarborough, in my presentation also, it was mentioned that the late Dr. Edgardo Gomez mentioned there were a lot of giant clams in Scarborough during his time. That's around 1980s. Right now in Masinloc, you can still find the restocking site we've established in the 20,000. So there are around 100 giant clams, Tridacna gaigas in that island in San Salvador. So some tourists visit that site, but they are not as well organized as in Samal. Although they have now Bantay Dagat that's helping protect the giant clam restocking site.
Thank you, Sir Patrick. Anyone from our RPs po na pwedeng mag-share? So, okay na po. Thank you, Sir Patrick. So, let's proceed to our next question. So, para po kay Dr. Patrick, what is the importance of clams in our oceans? As a president of an association, I want to know the details to relay to our fellow fisherfolk, for they can help to protect our resources.
Okay.
Sir Patrick.
It was presented again by a lot of author and the presenters that giant clams provide food and shelter to many organisms, invertebrates, and fish. So that's one of their main functions in the ocean. So it is important to protect them once they see giant clams. Although they're especially in many of the reef sites in the Philippines, their populations are still in very low abundance.
Thank you, sir. Sir, for these initiatives po na may na-involve tayo ng mga fisher folks dun sa concerned study sites nyo?
Yes, a lot of
Okay.
forest sites we involve fishers. Then maybe Sir Ma'am Girley can share her story in the involvement of fisher folk in the success of giant clam conservation.
Hello, good
Yes,
morning. In the case of Samal Island, the fisher folks, organized fisher folk, we call the Adicor United Fisher Folk Organization, were actually involved in the advocacy of the conservation of giant clams through marine protected areas. They're also involved in educating the tourists because they serve as tour guides. And they're involved in educating the tourists on the biology of giant clams, the species that are present in the area. So I think that's the involvement. And then they are actually involved in enforcing the MPA. And the turnover, they have the recruits that we have for giant clam, Tridacna derasa and Tridacna maxima, Tridacna derasa and Tridacna gigas, most of these are coming from the turnovers of fishers. That means they're already, we actually call them like community biodiversity champions at the level of the fisher folks, because they already know the importance of this species. And what is really good with the fishers is that they know that the species is recruit. That means it is not a common species. That's why they return it to the restocking site. This is not a common species. This is something new, and this must belong to the restocking site because they know it's Tridacna gigas and Tridacna derasa are no longer found many years ago. That's before the restocking, and that was actually validated during the FGD. So they know these are new and they return it. And the rare species, that's why in my presentation, it was clearly shown that Tridacna crocea and Tridacna noe were turnovers, and they're not found in the wild anymore. So when they say this is rare, they believe that they will live and they will be able to propagate once they will be placed in protected areas. And I think this is the role of the fishers in the case of Samal Island. But I cannot say that Samal Island is perfect. That is in the case of Adicor. Other areas, there are still poaching that is happening. So with regards to the fishers, it's not only local advocacy that they're doing. It's actually national and international, considering that the visitors of Tacloban tours are not only specific to the locality, but it has international tourists. So that like it has a multiplier effect. And I think that's the role, no, the contribution, the big contribution of our local fishers in Samal Island.
Thank you, ma'am.
May I add?
Ay, sige po, sir.
Okay. I-add ko lang doon sa yung importance ng giant clams sa ecosystem. Kasi po, kung maraming giant clams, if there are plenty of giant clams in the area, that would also invite a lot of fish. So, yung fishermen natin matutuwa kung maraming giant clams. Kasi the more giant clams you have, the more fish you have. Kasi yung giant clams nga, sabi kanina doon sa presentation, ay nagbibigay ng pagkain sa mga fish at substrate. Pwede rin silang kapitan ng mga iba't ibang marine life. At the same time, they add complexity to the reef. So the more giant clams, yung nga, kaya mas maganda kung maintindihan ng mga fishermen natin na pag mas maraming giant clams, mas marami silang makuhang isda or makitang isda sa lugar. Thank you po.
Thank you, Dr. Dolorosa. Yun nga po, isa sa benefit talaga ng giant clam is yung pag-enrich ng biodiversity po ng isang reef area. So ito po yung kagandahan pag naprotektahan natin yung giant clam, makakatulong po ito hindi lamang sa pag-enrich ng marine ecosystem, kundi yung livelihood din, yung livelihood din po ng mga fisher folks in the area. So yun po. This time po, magpipick po tayo nung last question na po natin. So ito po ay para kay Dr. Dolorosa. What can be done to more effectively prevent poaching and overharvesting and capturing the people who commit such crimes?
Thank you for that question.
Okay.
I think that would still boil down to education. Kinakailangan pa rin natin i-educate yung lahat-lahat ng mga tao. That's what we're doing. We're trying to reach more people, telling them the importance of giant clams. So kung mas maintindihan ng mga tao kung ano yung importance ng giant clams, they would stop harvesting the giant clams. Kung alam nila na threatened na sa or endangered na sa Palawan or sa Philippines yung giant clams, most of the species are endangered na. So they would help protect the giant clams instead of exploiting them. In addition to that, I think our law enforcers dapat medyo aktibo din. Kasi minsan parang nawawalan ng respeto o nawawalan ng, ano tawag natin yan, tiwala yung mga tao sa ating mga law enforcers. So dapat yung ating mga law enforcers should also maintain yung kaning good records when it comes to, apprehensions and all that. Kasi halimbawa lang sa Palawan ngayon, ang dami-daming taklobo na shells na confiscate, di ba? Pero ang tanong doon, nasaan na ngayon yung mga shells na yon? So, binalik ba yon doon sa dagat or dinala somewhere? At ano na yung status doon? So maraming mga haka-haka yung mga tao, so which we could not answer as well. In addition to that, ano pa ba? So, siguro tulong-tulong, lahat-lahat. There's a need for us to work together to ensure that we could protect and avoid the overharvesting or exploitation of giant clams so that in the future, in the near future, we could restore back the lost populations. I don't know kung may gustong idagdag yung iba kong mga kasamahan.
Thank you, sir. So yun din po ang hinihingi namin na statement din po as part of our advocacy dito po sa conservation and management ng giant clam. Ano pong masasabi po ng iba nating speaker? Pwede po muna kay Sir Patrick. Sir, kung meron po kayong i-share.
Ano ulit yung question?
Ano lang po masasabi nyo or may share nyo para po doon sa masustain po natin yung advocacy natin dito sa movement for the conservation and management po ng giant clam.
Okay, now we've in a lot of restocking sites, nakikita natin na yung iba successful, yung iba hindi ganun ka-successful. Maybe yung hindi masyadong successful, they still have to learn from the other sites. So yung hindi successful, for instance, we have this system in the Philippines that every so many years nag-iba yung government staff. So for instance, in one site, 10 years ago, we had a really good restocking site in one of the sites, one of our restocking sites. And then nagbago yung staff. Iba yung programs nila. Now they're not really protecting their ocean, their sea, yung reefs nila. So nagiging nawala yung magandang efforts. No, so that's one of the things we really need to look at kung paano natin i-address yan. Not just barangay level, pati town level, provincial level, maybe national level also. We know that in some sites, ang ganda ng support ng province, kaya maganda yung hanggang ngayon nasusustain yung program, yung restocking sites. And now in some sites, nakikita natin na kahit successful din yung pag-protect, dumadami yung mga giant clam babies, yung mga recruits, but still we can see na hindi siya enough kasi konti lang yung nilalabas na larvae, konti lang yung successfully nagsisettle. So there's still need to really culture and rear giant clams to increase yung stocks, especially in sites where there's a functioning hatcheries. So yun yung isa sa mga pwedeng tingnan in future. Kasi right now we have 8 sites with functioning hatcheries. I think we can still increase this number in the near future. This will increase also our chances of harnessing better yung socio-economic benefits ng giant clam conservation.
Thank you, sir. Si Dr. Girley naman po, ma'am, baka may last statement po kayo na pwedeng
Okay, so
i-share.
this is just in support to our program leader, Dr. Patrick Cabaitan. So there's a need for involvement of all stakeholders. So that means LGU, academic institutions has really a very important role in this aspect, considering that the academe is neutral, no, not politically inclined. People's organizations who are on the ground, and even the barangay level. And I think there's a need for strong governance and enforcement across leaderships at the LGU. Yun ang sinabi ni Dr. Patrick kanina na yung iba, umiiba yung agenda pag nag-change na yung leadership. In the case of Samal Island, for example, I can say, and in the case of DNSC, we have strong support across leaderships. And I think whether political or in the academic, I think this is one factor that made it a success in our area. Hindi po nagbabago yung suporta nila sa Tacloban. Yun lang po. Thank you.
Thank you, Ma'am Girley. Si Ma'am Conaco naman po. Ma'am, if you have last statement po.
We all agree essentially that it's really important to have all stakeholders working together. We've seen that in the best sites that we've been to, na interested talaga lahat sila. They're all interested to learn about their clams. They all want to continue monitoring, and their support for the people who want to do this from their local government units. To add to that, I think it's also nice na for a lot more, it's nice that a lot of people from different places around the Philippines are watching today. And I do hope that this will spur interest in monitoring their sites. Because one of the nice things we found through this program is ang daming clams. They're very pretty, they're very beautiful in all over the Philippines. It really just highlights the resilience of some of these reefs, despite all the disturbances na nandyan. And you can imagine how much more beautiful they will be if we continued protecting those. And in my case, because we're interested in looking at genetic resources, I'm hoping that this is something that will continue to push through, because we do want to learn more about our own resources of giant clams. We want to know how diverse they are, whether we can culture them more. And it's good that there's a lot more hatcheries now. So I think concerted yung efforts ng protection, ng research, and conservation. And I think it's really all of these things coming together that will boost the success of giant clam conservation. Thank you.
Thank you po, Dr. Conaco. Maraming salamat po sa ating mga resource persons. At dyan na po nagtatapos yung ating Q&A. Salamat po sa mga aktibong paglahok sa ating question and answer. At sa mga hindi po natin nasagot na mga katanungan, we will forward this to the research team po or sa concerned resource person. So and now, nalalapit na po yung pagtapos nating programa. At may konti lang po kaming paalala. Sa ulitin po, maraming salamat po sa ating mga resource person. So mamaya po, ipipin po namin yung CSF dito po sa comment sections. Okay? And now to close our program, let's hear a closing remark to be given by Dr. Marian M. Asidera, the Director of Marine Resources Research Division.
Salamat, JP, and a pleasant day to everyone. I would like to extend Picar's appreciation to our great speakers. So we have Dr. Cabaitan, Dr. Dolorosa, Dr. Gumanao, and Dr. Conaco, who shared the results of the program with us and provided insights on how we may move forward on the future giant clam conservation strategies and management in the country. Also, I would like to express my gratitude to all of you who joined and participated in this activity or in this webinar. I look forward to having you in our future webinars. This activity would not have been successful without all of you. I believe that the presentations we have gained a better understanding on the current state of the giant clams restocked for around 40 years ago in the different areas of the country. Now, we have the scientific data to support anecdotal accounts that the restocked giant clams, Tridacna gigas, indeed grew to maturity. And the sightings of natural recruits observed indicate that the restocked giant clams contributed to replenishment of our local populations. We have the initial information to help us recalibrate protection and conservation strategies for our giant clams. And our understanding on the socio-economic implications of giant clam diversity is enhanced, especially in tourism and community-based farming. And that 8 out of the 12 giant clam species in the world are still in our reefs. We have increased the interest of stakeholders to engage communities in giant clam conservation through the feedback mechanisms conducted throughout the duration of the program and through this webinar. We have also increased and then let us all continue to protect our giant clams, not only for the marine environment, but not only for us, but for our local communities and for the future. So once again, thank you very much. It has been a pleasure to be with all of you today. See you all in our future webinars. Keep safe, everyone, and God bless po. Maraming salamat po.